The next disorder of the endocrine system we're going to talk about relates to malfunctions in the pancreas. And one important thing to remember about the pancreas before we really get into looking at the specifics of diseases of the pancreas is that the pancreas is one of those few organs in the body that has both exocrine and endocrine function. And you'll remember endocrine function means that relates to the manufacture of a hormone and its release and eventual circulation in the bloodstream. Exocrine function means the release of a substance such as an enzyme, a digestive enzyme in the case of the pancreas, that gets released into a luminal opening such as the small intestine. So before we get started on talking about diseases of the pancreas, I want you to just take a minute and see if you can for yourself think of at least 2 different endocrine products that the pancreas manufactures, and think of at least 3 exocrine products that the pancreas manufactures. And go ahead and hit pause right now. See if you can write them down, and then I'll talk about some of the different types of products that are made in just a second. So let's see what you came up with what ideas you came up with in terms of endocrine products that the pancreas secretes. Of course, the most obvious of them is insulin. And insulin is the hormone that is responsible for decreasing blood sugar levels by inducing different tissues in the body to take up glucose and remove it from the blood. And on the next slide, we're going to look a little more in depth at that. Insulin, you'll remember, is secreted by the beta cells in the pancreatic islets. Another common type of hormone that the pancreas secretes is called glucagon. Glucagon is secreted by the alpha cells in the pancreatic islets, or the islets of Langerhans, if you want to call them that. And glucagon is antagonistic to insulin. It does the opposite. So glucagon's principal job is to elevate blood sugar levels when needed in the body. And there's actually then also a third hormone you may not remember that the pancreas secretes, and that is somatostatin. Somatostatin. That's released by the delta cells of the pancreatic islets. And those delta cells that are manufacturing this somatostatin are doing so to help modulate the secretion of glucagon and insulin. So basically, it helps to fine-tune and adjust when the beta cells are more active, when the alpha cells are more active, based on how much sugar is in the blood, based on intake of fatty acids and amino acids, etc., from the meals that you eat. So somatostatin is sort of the modulator for insulin and glucagon, and it acts actually as a paracrine. Because it only influences the alpha and beta cells, it doesn't actually get released into the blood and circulate around the body the way insulin and glucagon do. Now, what about exocrine products? Well, the pancreas secretes lots of different types of digestive enzymes that are really critical for helping you do the ultimate process of your food to get it small enough to be absorbed in the body. So I'll throw out a few of them. You've got carboxypeptidase, which is an enzyme that helps degrade amino acids. You have pancreatic lipase to help digest fats. You have pancreatic amylase, which assists in the digestion of carbohydrates. You have trypsin and chymotrypsin, both of which also assist in the digestion of proteins into individual amino acids. And what else am I missing here? Oh, yeah, the 2 ribonuclease for digesting RNA and deoxyribonuclease for digesting DNA. So you can see that the pancreas secretes a whole lot of different exocrine products and relatively a lot fewer endocrine products, but they're both critical for proper functioning of digestion and of modulation of sugar levels in the body. Now, if we look at the specific effects of insulin, we can see that it has different kinds of impact on different tissues in the body. And I want to take you first through this diagram here off to the right before I talk to you about the different types of diabetes mellitus. So here's my insulin. Imagine insulin secretion climbs, such as after you've eaten a meal. And I want to show you how this insulin influences different tissues. One of the major tissues that responds to insulin in the body is adipose tissue or fat tissue. And it has a number of different ways in which it responds to insulin. When sugar levels in the blood are high and insulin is released into the blood, adipose cells respond to that insulin by increasing the amount of glucose that they take up from the blood. So, in other words, storage of glucose. They then take that glucose and begin converting it into fat in a process called lipogenesis, which literally means the generation of new lipid molecules. At the same time that we're getting an increase in the amount of sugar that the adipose tissue is storing and the amount of lipids that the adipose tissue is manufacturing, we're also getting a decrease in lipolysis. And lipolysis, you can figure out what that means. Lysis means to split apart. Lipo refers to fats. So in other words, we're getting a decrease in the amount of fats that are getting broken down and released into the blood, which should make sense. If at the time we're manufacturing new molecules of lipids, we don't want to be breaking them down right away. So the overall impact of insulin on adipose tissue is that these cells get larger. Simply because they're taking on more sugar and converting it to more fat, and we get more lipid uptake. We're going to find out later that that is a critical component of actually feeding into the development of diabetes mellitus. The more fat tissue you have, the more susceptible you are to developing diabetes mellitus. What about skeletal muscle tissue? Well, if insulin levels go up, skeletal muscle tissue has a little different, and this is also for cardiac tissue as well. Has a different way of responding compared to fat tissue. When insulin levels go up, that's because sugar levels are up in the blood. That muscle tissue will take up that additional glucose and will begin converting it into the stored version of glucose called glycogen. Glycogen, you'll remember, are those long strings or chains of glucose that are highly branched. Okay, they're like the human version of starch that plants make. It's basically the same idea, it's just a little different configuration of the stringing together of the glucose molecules. Now, because I have more sugar uptake, that means I have more energy available to fuel such things as synthesis of new proteins in that muscle tissue. So that's yet another way in which muscle tissue, striated muscle tissue, responds to insulin. In the liver, we have yet a different suite of responses to insulin. Insulin levels climb when blood sugar levels climb, and as a result, the liver will begin to take that extra sugar from the blood, and just like the muscle tissue, it will begin to synthesize and store glycogen, take all those sugar molecules and string them together in long chains. It will also, because of the increase in the amount of sugar present, also, like the fat tissue, begin to manufacture more lipids. So we'll get lipogenesis, or the generation of new lipid molecules. You will also see in the liver, when sugar levels are high in the blood, you will see a decrease in the amount of gluconeogenesis occurring. Now, if you take that word apart, you can figure out what it means. Genesis means to generate or create. Neo means new. Gluco refers to sugar molecules. So we are literally generating de novo, or from scratch, we're generating new sugar molecules. And that process declines. That should make sense. The liver shouldn't be making new sugar groups if there's already so much sugar in the blood as it is, that it's doing everything it can at the moment to just store what's already available. So there's no sense in making any more. So those are the principal impacts of insulin on those three different target tissues. In general, for other body tissues, the insulin has the impact of encouraging glucose uptake. Now, if I have some sort of a perturbation to any of these processes, it can lead to the development of disease. And in the case of insulin, the disease that can result if the body isn't responding properly to insulin or insulin isn't being made properly, we can get what's called diabetes mellitus. And there are two main types of diabetes mellitus, and you probably know this already, at least in general. Type 1 diabetes results when the body is unable to manufacture sufficient amounts of insulin to respond to the glucose levels in the blood. Gestational diabetes, which I'm not going to talk about, but just so you know, gestational diabetes is sort of a subtype of type 1 diabetes mellitus that occurs only during pregnancy, but it's often a sort of a warning to the woman that she is at much higher risk of developing diabetes at a later date if she has gestational diabetes during pregnancy. We're going to talk in a little bit about how exactly it comes about that the body loses its ability to make insulin in type 1 diabetes. For now, just know this is generally speaking the type of diabetes we see with onset in childhood rather than adulthood. In fact, it's often called juvenile diabetes mellitus because that's most often, if you're going to develop type 1 diabetes, you usually have developed it by the time you're about 12 years old. The second type of diabetes accounts for roughly about 95% of all cases of diabetes mellitus, and that's type 2. Type 2 diabetes mellitus occurs if the target cells, these guys here and here, that are supposed to be responding to insulin, do not do so appropriately. So by definition, type 2 diabetes is basically an inability of target organs and target cells to respond appropriately to insulin. This is also often called adult onset diabetes mellitus because it's typically seen in people in their 30s or 40s, although in the United States it's becoming more prevalent among people in their 20s, and even some people in their teens are beginning to develop type 2 diabetes mellitus. As a sort of a sequelae of this, as a follow-up, in sequel to the development of type 2 diabetes, we often start to see beta cell malfunctioning. And we'll talk about that in a little bit in more detail. So how do we diagnose diabetes mellitus? Well, the principal way, the starting point way, is to, for example, look at the levels of glucose in the blood, and also to look at the amount of glucose that's found in the urine. So, for example, if we look at what the normal range, I guess, if you would say, of what glucose levels should be in a normal, healthy person, plasma levels of glucose in the blood should be somewhere around 70 to 120 milligrams per deciliter of blood serum. If a person is going to be diagnosed with diabetes mellitus, they've got to have one of these happening. Either on a random draw of their blood, multiple times they get a value over 200 milligrams per DL for the amount of sugar in the plasma, or if they do what's called a fasting glucose test, where they have the person go for a period of time without consumption, and then they give them a test. If they have a value of 126 milligrams, or higher more than once, or there's also something called an abnormal oral glucose tolerance test, which I'm not going to go into, but if they get an abnormal value for their oral glucose tolerance test, that can also be indicative of diabetes mellitus. Now, I also said that glucose in the urine, or glucosuria, is another indicator of diabetes mellitus. In a normal person, there should be no glucose whatsoever present in the urine. In a person with diabetes mellitus, they may have very high levels. In fact, diabetes mellitus literally means sweet urine. Mellitus means sweet. Diabetes means to run through In other words, the formation of urine, the fluid that is run through the body and come out the other side, more or less. So this is sweet urine, and indeed, in the 1800s and early 1900s, before they had sugar test material for urine, the way that doctors diagnosed diabetes mellitus was to taste the patient's urine to see if it was sweet due to the presence of glucose. So as I said before, we have two main types of diabetes mellitus. And type 1 diabetes mellitus, or juvenile onset diabetes, is by far the rarer type. Only roughly about 5% or so of all diabetes cases are type 1 cases. Here's how we think that diabetes type 1 develops. We know it's an autoimmune disease, and it represents a type of hypersensitivity reaction in which we think there are either helper T cells or cytotoxic T cells, or both, that become sensitized to either a receptor found on the surface of pancreatic beta cells, the ones that secrete insulin, or that become sensitive to the insulin itself. And as a result, they initiate an abnormal immune response. They recruit, you know the story, they recruit macrophages to the area. The macrophages begin to secrete all those different types of damaging cytokines we learned about earlier in the semester. B cells get activated, the cytotoxic T cells are activated in cloning, and we get this massive attack of the beta cells in the pancreas and their ultimate destruction. What causes this? Well, that's a huge area of debate still in diabetes research. We know genetics play a factor. People that have a particular type of HLA2, which is the same as an MHC2, by the way, receptor on their surface, their cells tend to be more susceptible to many different types of autoimmune diseases, things like Hashimoto's syndrome, Graves' disease, Addison disease, and also diabetes mellitus 1. But we also think that there's probably some sort of infectious agent. Maybe it's the Epstein-Barr virus. It could be mumps virus. It could be cytomegalovirus. It might even be, there are some people that think it might be bovine insulin. In other words, the insulin that's found in the milk that we drink in childhood that may, in some people, if they're genetically susceptible, trigger this sensitization of CD4 and CD8 cells and initiate the destruction of the beta cells in the pancreas. These are all speculative. We don't have the definitive answer about what causes this. All we do know is that it is an autoimmune disease. This is a type of hypersensitivity reaction. And what I think is most interesting about this is that in order for somebody to actually become overtly or diabetic, in order for them to finally develop the sort of clinical signs and symptoms we associate with untreated diabetes mellitus, they have to have had somewhere between 80 to 90% of all of the B cells in their pancreas destroyed. What that says is that you can function on a relatively low number of beta cells and still have completely normal blood glucose chemistry. It takes a huge hit to those beta cells before you actually develop the kinds of conditions like diabetes that are associated with having hyperglycemia. Okay, now just so you know over here, if you were interested, I wanted to show you kind of here the sequelae, that means the sequence of events that occur as a result of destruction of the beta cells in the pancreas. Obviously, when the beta cells become destroyed, I'm not making insulin anymore, so my sugar levels in the blood climb, and I also find out that most of the cells in my body are now not able to access that glucose. Okay, cells in the body, most somatic cells, have to have permission from insulin before they can begin to take up sugar. So if I stop making insulin, well, those cells they're sitting around waiting for permission to take up all the sugar that's in the blood. And so in essence, what happens is those cells begin to starve to death. And that triggers off a whole cascade of responses. We get a massive wide announcement of the body that these cells are starving. We need sugar. And so the body's way of responding to that is to do whatever it can to elevate blood sugar levels, thinking that that's going to solve the problem, that because the cells are starving, the only way to fix it is to provide them with glucose. And of course, ironically, that doesn't do any good at all because there is plenty of glucose already present in the blood. It's just that the cells cannot access it because there's no insulin available. So if we look at adipose tissue, the adipose tissue begins to break down the lipids and release convert them into free fatty acids and glycerols and release those into the blood, where they then travel to the liver, and the liver then has the capability of manipulating those lipids to produce new sugars, gluconeogenesis. As a side effect, this causes an elevation in the bad type of cholesterol, an elevation in LDL cholesterol levels, and a decrease in the good kind of cholesterol, the HDL levels. And this leads to the development of things like atherosclerosis, the development of plaques in arteries because of the elevated cholesterol levels. We'll talk more about that later. It also, because we're making more glucose through gluconeogenesis, we're making the hyperglycemia of the blood even worse. Meanwhile, the muscle tissue is also starved for sugar. So it relies on its glycogen stores. Remember, muscle tissue stores sugar in the form of glycogen for later use. It breaks down all that glycogen to utilize as an energy source. It also begins to break down its own proteins to use that as an alternate energy source since it cannot get access to sugar. So that causes an increase in the amount of amino acids that are being released in the blood. The amino acids go to the liver, and there they are also used to produce new sugar molecules. And the end result is I get muscle wasting. I get lots of fatigue because, of course, if you think about it, what's your body going to be doing if it can't access sugar? All your body cells are going to be starving for sugars. You're going to experience fatigue. You're going to experience some of these symptoms that I'm going to talk about in a minute called polyuria and polydipsia. And if it goes untreated, we can eventually progress to a state of death. And this actually used to happen to people prior to our development of exogenous sources of insulin to give people. There was no way to treat it prior to that point, and so people would literally waste away to nothing and die. It's a pretty awful disease.